Hello, good afternoon. Welcome back to Virology. Today we're going to look at how to make RNA from RNA. This is a unique process because most things on the planet have DNA. Many viruses have DNA and just some have RNA. So those are the viruses we're going to talk about today. A little bit of RNA history. In 1935, Stanley crystallized tobacco mosaic virus. He made crystals of it. We talked about this before. And he finds that the crystals contain 5% RNA. And he thinks it's a contaminant. He thinks that the protein is the genetic material. He got a Nobel Prize for crystallizing TMB. He probably should have given it back for concluding that the RNA was a contaminant. Now, in 1944, DNA is found to be the genetic material in bacteria. Famous experiment at Rockefeller. Oswald, Avery, and McLeod showed that you could change the property of a bacteria by putting DNA into it. So DNA is genetic material, at least for bacteria. In 1952, the Hershey-Chase experiment, which we talked about, showing that the DNA of a bacteriophage is genetic material. 1956, Frankel-Conrad tobacco mosaic virus, RNA is genetic material. Okay, so by the late 50s, RNA is fully respected as genetic material. It begins to be identified in many animal viruses. And 1960s, people start to study it because they want to know how it replicates. Because cells don't have RNA as genetic material. They have RNA in other forms, right? But they're not the genetic material. So today we're going to talk about viruses with RNA genomes. And in the Baltimore scheme, that will be group 3 double-stranded RNA viruses, group 5 negative-strand RNA viruses, and group 4 plus-strand RNA viruses. The group 6 plus-strand RNA that go through DNA intermediate is a separate electron on its own. Very interesting set of viruses. But they do have RNA as the genetic material. It's just that they go through DNA intermediate, so we consider them separately. Now, the first identification of enzymes that can make RNA from RNA was in an experiment like this. You take cells. This is a cell. This is not an egg. It's a cell. And you infect it with a virus, in this case, poliovirus. And then you incubate it for different hours post-infection. And at different times, you break the cells open and you add the 4 triphosphates, one of which has a radioactive label. And then you measure RNA synthesis. And what you see is shown on the graph on the right. So first, these are hours post-infection on the x-axis. There are 2 y-axes. The y on the left is RNA polymerase activity. And then the one on the right is infectious poliovirus. And those are squares. So you can see, between 2 and 3 hours, the amount of poliovirus rapidly increases. So the cells are infected. And at around the same time, you get an increase. These circles are the synthesis of RNA by this extract from the infected cells. So there's some kind of RNA polymerase activity in infected cells. It's the first time that it had ever been seen. Of course, people knew that DNA could be copied, that DNA could be copied into DNA or RNAs of various sorts. But people didn't know that RNA could be copied into RNA. The next step was the finding that RNA polymerase was found in particles of negative-strand viruses. Remember, negative-strand viruses have the opposite of mRNA. The RNA can't be translated as soon as it comes into cells. And so the logic was there must be a polymerase in the particle to make mRNAs once the particle gets into cells. And so that was discovered in particles of minus-strand RNAs. Plus-strand RNA viruses, for the most part, do not need to carry polymerases, right? Because their RNA can be translated into protein as soon as it gets into cells. Then in the era of recombinant DNA and sequencing, we were able to see certain amino acid motifs like GDD, gly, asp, asp, that were signatures for the active site of an RNA polymerase. We're able to make recombinant proteins that show they have activity and solve molecular structure. So we've come a long way. And so this idea of RNA polymerase in the virus particle is very important for you to conceptualize, right? So we have some minus strand viruses, right? Like VSV and influenza virus. These contain an RNA polymerase. The RNA is coated with protein. It's a nucleocapsid, and the polymerase is in with it, because as soon as that RNA gets in the cell, it has to be made into messenger RNA. We'll see that today. Plus-strand genomes, for the most part, have no RNA polymerase. They're the exceptions, and they're naked RNA. There's no nucleocapsid, but there are exceptions. The retroviruses and the coronaviruses do have nucleocapsids. They have RNA protein complexes in particle. And the retroviruses, of course, even though they're plus-stranded, they have an enzyme in the particle, reverse transcriptase, that copies the RNA to DNA. So it's very different from what we're talking about today. That's why we're going to consider them separately. But for the most part, viruses with plus-strand genomes do not carry a polymerase in the particle because the RNA can be translated as soon as it gets into cells. And examples would be poliovirus, flaviviruses, SARS coronaviruses, or any coronavirus, retroviruses, and retroviruses. Now, the final set of RNA viruses, double-stranded RNA genomes. The RNA is not a nucleocapsid, it's naked. But it's double-stranded. And there is a polymerase in the particle, because double-stranded RNA cannot be translated. You have to remember that. Double-stranded RNA, if it gets into a cell, ribosomes cannot bind to it because it's a double strand. The plus strand is occluded. So there is a polymerase in the particle that will copy that double strand and make messenger RNA. We will see all of this later today, okay? Just a reminder about nucleocapsids. It's basically, a nucleocapsid is a nucleic acid coated with protein that has got something else wrapped around it. That's why it's nucleo, right? To give you this idea of nucleus. A capsid is just the shell with nucleic acid in it. And if that nucleic acid had protein bound to it, it would be a nucleocapsid, giving this idea of a nucleus, all right? So these are examples of nucleocapsids, VSV. Again, protein, many copies of the same protein bound to the RNA. Influenza virus, many copies of the same protein bound to RNA. And these are the structures of these two nucleocapsids here on the bottom. This is the actual three-dimensional structure. Here is a monomer of the nucleocapsid. There's the RNA in green binding in this groove. And many of these line up. So here, you can barely see it in dark blue. There's a monomer of the nucleocapsid. They all line up next to each other to form this helix, and the RNA threads through them. And the same thing with influenza virus. So those are nucleocapsids. RNA is not a line on a slide, as I often show. It has extensive secondary and tertiary structure in the form of stem loops where the RNA can bind to itself, and they can be very extensive, as you can see here. And here at the end is something called a pseudoknot. So this is a stem loop where bases in the loop base pair additionally with bases downstream, and they form this configuration which, here's the three-dimensional structure of it. It looks like a knot, but it's not. So we call it a pseudoknot, all right? And here's just an example on the right. This is the leader sequence of HIV-1, many stem loops which form highly structured regions. And all of these are biologically important. They have a role to play in replication. Okay, the rules for RNA synthesis. That's a piece of RNA that's a line, which is wrong. I just told you it shouldn't be a line, but I'm going to draw lines a lot. First rule, the RNA genome has to be copied from end to end with no loss of nucleotide sequence. That may seem obvious, but we'll see some examples where this is not happening. So you have to copy the whole genome, because if you lose sequence at the end, it's not going to be functional. And secondly, from this viral RNA, you have to make messenger RNAs that the cell can translate to make proteins, right? Now, sometimes the genome is the mRNA, but not always. Sometimes it's not. If it's negative-strand, it's not. And even for some plus-strand viruses, it's not mRNA. Like retroviruses, the plus-strand RNA is not messenger RNA. It's not translated when it gets into the cell. All right, so these two rules, very important. Okay, more rules for RNA-directed RNA synthesis. So RNA synthesis begins and ends at very specific places on the template. So here's a template shown here in gray, and here is a primer. Sometimes RNA synthesis requires a primer, sometimes it does not. When it doesn't need a primer, we say it initiates synthesis de novo. And cellular DNA-dependent RNA polymerases, the polymerases that make mRNA, they initiate de novo. But sometimes RNA polymerases are like that, but sometimes they require a primer as well. Here, we're showing an RNA polymerase needing a primer in red. It base pairs with the template, and then the RNA polymerase adds NTPs to the template, to the product. The template is read in a 3 to 5 prime direction. The product is added in a 5 to 3 prime direction. Okay, so synthesis is 5 to 3 prime. The template is read in a 3 to 5 prime direction. And not all RNA synthesis is templated. Sometimes there's non-templated RNA synthesis. We'll see some examples of that. All right, now, initiation of RNA synthesis. Again, you can have de novo initiation, which means you don't need a primer. That's shown at the top. So here's an RNA that has to be copied, and the polymerase can simply add bases to the 3' ends. Right, it would add this one and the next one and so forth. Then sometimes we need primers. And that's shown at the bottom here. Here we have a protein primer where the base is linked to a protein, and that is the first base that's put down, and then subsequent bases are added. And sometimes the primer is a capped piece of RNA from the cell, which again serves as a primer to allow RNA synthesis. Now this one on top here, just to be clear, N1, N2 are the bases in the genome that needs to be replicated. And we're adding NTPs one by one to make a copy. And so there's no primer involved here. That's the point. Whereas at the bottom, you need either a protein primer or a nucleic acid primer. Now, the way you make RNA is by what is called a two-metal mechanism of catalysis. This is really metal, this process. Maybe not all of you get that, right? It's great. This is actually the same that happens with DNA. So I'm going to go through it with you. We have a template, we have a primer, and we have a polymerase extending the primer. And so that's what we're seeing on the right here, except it's for DNA, but I use the same figure for both. Same principles. Here's the template all the way on the right, 3' to 5'. We have our riboses with bases, G, U, A, or U, or C, and they are linked by phosphate bonds. Okay, so that is the template that's going to be copied. And here we have added, the primer is up here, and we have added a base, this C complementary to G, so we've added C, ribose, phosphate. Here we've added an A, should be a U because it's RNA. And the T now is in the process of being added, so you can see what happens. So the T, each of these bases comes on as a triphosphate, right? With 3 phosphates attached. There's 1, there's 2, and there's 3. and they're called the alpha, the beta, and the gamma phosphate. And in the end, after the reaction, the beta and the gamma phosphates are gone, and what you are left with is the alpha phosphate. And so you just have one phosphate between each ribose, whereas the beginning triphosphate, there are three. Now, what happens in the active site of the RNA polymerase or the DNA polymerase is there are two metals that are being coordinated. In this case, it's a magnesium, it's a metal, and it's being held in place by specific amino acids, this aspartate and this aspartate. So there's two aspartates, and I'll show you these in the structure. Their job is to hold metals in place, and the metal, the magnesium, coordinates the nucleophilic attacks which get rid of the beta and the gamma phosphate. So you have nucleophilic attacks going on here that leave one phosphate and these two phosphates come off. That's coordinated by the magnesium. The magnesium holds the phosphates in place while this happens. So this happens in the catalytic site of the enzyme. This happens for both DNA and RNA, and that's why we call it a two-metal mechanism, because there's two metals involved. This can sometimes be manganese, but mostly it's magnesium. The first question is, which is a universal rule about RNA-directed RNA synthesis? A, RDRP, that's the RNA-dependent RNA polymerase, may initiate de novo or require a primer. RNA synthesis initiates randomly on the template. RNA is synthesized in a 3 to 5' direction. RNA synthesis is always template directed. So which is a universal rule? Okay, let's see how we did. 36 out of 40. The polymerase may initiate de novo or require a primer. That's correct. RNA synthesis initiates randomly. No, it doesn't. It always initiates at a specific place. RNA is synthesized in a 3 to 5' direction. No, it's synthesized in a 5 to 3'. It's copied in a 3 to 5', and it's always template directed. Sometimes synthesis is not templated. So we now have structures of all the polymerases. There are 4 different kinds of polymerase, as you can see here. We have the Clenau is a DNA-dependent DNA polymerase from bacteria. T7 RNA polymerase is a DNA-dependent RNA polymerase, makes transcripts from DNA. Reverse transcriptase takes RNA and makes a DNA copy, and then RNA polymerase, RNA-dependent RNA polymerase in the form of the poliovirus polymerase is shown there. So that's it. There are only 4 different kinds of nucleic acid polymerase, all shown here. And they all look pretty similar structurally. They have different sequences, but there are conserved spots. They all look like a right hand, as you can see there, with the active site in the palm of the hand. And for some polymerases, the fingers and thumb are closed. So you can see for polio polymerase, the fingers and the thumb close, and so they make a closed active site. But for the others, they are not really closed. Okay? In addition, there are conserved sequences. So at top are the actual amino acid sequences of these enzymes. And the colored parts, like red and yellow and green and so forth, show the conserved parts of the polymerases. So these 4 polymerases have several places that are very similar in sequence. So they must have most likely evolved from a common precursor. The most important one here for us to look at is yellow. Racaniello, yeah, that is the most important one. Right here, because that is the active site of the enzyme. And you can see it here on the 4 structures in yellow, 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 and yellow. And these have very conserved amino acids, in particular, gly-asp-asp. So those are the 2 asps that coordinate the magnesiums that I just showed you in the previous slide. So that's the poliovirus polymerase, that's the reovirus. A different double-strand RNA virus has ala-asp-asin, but same function. Influenza has serine-asp-asp, VSV has gly-asp-asp, and HIV has met-asp-asp. So all of those are slightly different, but they their function is to coordinate those magnesium metals so that catalysis can occur. And that's happening in the active site of these enzymes. So let's take a close look at the poliovirus polymerase, because we're looking at RNA-dependent RNA polymerases today. So here is a view from the front of the enzyme. This is just a trace of the amino acid backbone, so we don't have any side chains. I just want you to be able to see the yellow conserved sequence here. And those two, I put in 2 side chains for those 2 aspartate residues, right there and right there. So the whole yellow part is highly conserved, but these are the 2 aspartates right there. And again, this active site right here, which also comprises the red part, the red strand, that active site is in the palm of the right hand. And this enzyme is closed, and you can see the fingers and thumb domains are closed. They touch each other. So the active site is encircled. So in reality, of course, there are side chains in these enzymes, and this is what it looks like. I've taken away some side chains for clarity. I have 3 different views here of the polio polymerase. We have the top, we have the front, and we have the back. So the top shows you where the template comes in. So the single-stranded plus RNA comes in the top of the enzyme, and you can just see in yellow there, that's part of the active site. And there in magenta, those are the 2 magnesiums that coordinate, the 2 aspartates that coordinate the magnesium. We've colored them in magenta. So the RNA comes in, passes over the active site, so it kind of makes a right turn, and then exit on the side of the molecule. And of course, as the RNA is going through the active site, it's copied, so now what comes out is actually a double-stranded form. Okay, so that's where the template goes in. This is a front view, sort of to show you the same. Here's the template entry in the top. It's coming in single-stranded, and the exit is now in the front in your face there. So it goes in single-stranded and comes out double-stranded. And finally, the back of the molecule. Why am I showing you the back? Because that's where the NTPs go in, right? So the RNA goes in, and it flows over the active site, but it needs NTPs to be copied, right? The 4 NTPs. There's a channel in the back of the enzymes, in the enzyme where they go in. So it's called the NTP entry channel. So that's the overall structure of poliovirus RNA polymerase. The other ones are similar. They have some little details that are slightly different for different functions. But basically what I want you to know is that the template comes in, it passes over the active site where bases are added, and then it comes out double-stranded. Now, you may wonder, how does it know what base to pick, right? Because there's a base at the active site ready to be added. The complement is ready to be added. So let's say it's an A. How does the enzyme know to bring in a U? It doesn't. It tries all 4. They all 4 come in really fast, okay? One comes in, it's wrong, it goes out. One comes in, it's wrong, goes out. The right one comes in, boom. But sometimes it puts the wrong one in. It's a bit lazy. And that's where that's where mutations of RNA viruses come in. And that gives us all the diversity. That's why we're worried about H5N1, because at every RNA cycle, it's making mutations. And we're worried that one will randomly arise that allows it to transmit in people. It always mutates. Every replication cycle, it mutates because sometimes the polymerase is sloppy. Okay. Now, I want to go through the specific mechanisms of copying these genomes. So we're going to start with plus-strand RNA viruses. We're going to focus on the picornaviruses and the flaviviruses. They have a single strand of plus RNA. And that plus RNA, of course, when it goes into a cell, can be translated into a protein, many proteins. Among the proteins made are the RNA polymerase, which will then copy the plus into a minus strand. The only function of that minus strand is to make more plus strands. That's it, nothing else. It's copied to make plus strands, and those can be translated or they can be packaged into virus particles. The alpha viruses are also plus-strand viruses like Sinbist, Semliki forest, chikungunya. They're plus-strand viruses with the twist is that there's a short mRNA. So the whole genome is plus-strand, but it's not all translated. There has to be a short subgenomic mRNA made. Just a little twist on the same strategy. All right, so how does this work? For poliovirus, the particle attaches to a receptor. The RNA comes out of the particle into the cytosol. The RNA is then engaged by ribosomes, right? It's a plus-strand RNA. It can be translated directly into proteins. So we have a variety of proteins made, including proteins that induce the formation of these vesicles, these membrane vesicles, and it's on those that RNA synthesis occurs. Pretty much all RNA viruses synthesize RNA on vesicles. We're not sure why. We have a lot of ideas, but they all pretty much do. And so you can see here, we have the plus-strand RNA that came in, and on the vesicle, it's copied into double strands, all right? And then from those, you can make more plus strands. And among all the other proteins made are the capsid proteins, which come together with the RNA and make new virus particles. So that's an overview of RNA synthesis. It's relatively simple. The genome is the same as the message for this virus. The genome is mRNA. Sometimes it's translated and sometimes it's packaged into virus particles. So here's the genome just to show you what goes on. It's a plus-stranded RNA, about 7.5 kb long. It's translated into a long protein called a polyprotein, and this has proteases embedded in it that chop it up and give you capsid proteins. Here on the left, VP1, 2, 3, and then on the right, all the other proteins. These are all involved in RNA synthesis, and here's the RNA polymerase 3D Pol. So this is an interesting strategy. It makes a long protein and then cuts it up with proteases. Other viruses do this as well. Now, at the very 5' end of this RNA, there's a protein linked to it covalently called VPG. And here's a diagram that shows how it's linked. So here is the very first base of poliovirus genome RNA, this U. It's part of a very highly structured RNA region called a clover leaf. And this is connected. Actually, the last base is here, this uridine. So that's the penultimate U. This is the last U. And that last U is connected to a small protein by a phosphate bond. So that protein called VPG is about 21, 23 amino acids long. This is the primer for RNA synthesis. So when the RNA is being made, a few bases are added to VPG, and then they're extended on the template. So that's the primer. Poliovirus RNA synthesis requires a primer. The genome actually has extensive secondary structure. It has that clover leaf at the 5' end that I told you about. In the middle is what's called a cis-acting RNA element. This is where the primer is made, VPG is made. I'll show you in a moment. That's actually movable. You can put it anywhere in the genome as long as you don't mess up the translation. And then at the 3' end, we have a pseudoknot. And we think these signals ensure that cellular RNAs are not copied, because cellular RNAs are never copied by the poliovirus RNA polymerase, only poliovirus RNAs. And we think these signals play a role in that. Okay, so this now shows you how VPG is made into a primer. So the Cre element is that little stem loop I showed you in the genome. And a couple of molecules of polymerase bind to this stem loop. And then VPG comes in, and the polymerase adds two U's to it. So this loop here at the top of Cre is full of A's, and so they serve as a template for addition of U. So VPG is a protein. We add UU, two U's to it. This is now going to be the primer for RNA synthesis. And this happens on membranes. So here's the viral RNA. Here is the 5' clover leaf-like structure. And there's VPG on the plus-strand RNA. The RNA is actually circularized. The poly-A tail binds a cellular protein called poly-A binding protein, which in turn binds the clover leaf structure. And then a molecule of VPG comes in with PU-PU and starts priming at the poly A. So the VPG-PU-PU is made at this crease sequence. And how it gets there, from there to there, we don't know. All we know is that circular RNA is important. Somehow it goes from the CRE to here, and then the two U's bind to two A's at the 3' end of the RNA, and then the polymerase begins to make a copy. And then you get a minus strand and end up with a double-stranded template. And the same thing happens when you make the other strand. Okay, next question. Which is a part of the poliovirus replication strategy? Production of subgenomic mRNAs, de novo without primer initiation of RNA synthesis, circularization of template for initiation of RNA synthesis, all of the above. All right, let's have a look. Which is part of the polio replication strategy? Subgenomic mRNAs, no. I'll show you the figure. Without primer initiation, no, VPG is the primer, right? I went through a lot to show you that. Circularization of the template, yeah, that's needed. So it's not all of the above. So let's take a look at what we have here. So what's the first one? The production of subgenomic RNAs. No, where's a nice slide that shows that? Now here. So we have the plus RNA that comes in, it's made into a minus RNA, and then another plus. Maybe you confused it with alpha viruses, which is a different family. They make subgenomic RNAs. I'm sorry. That's not the right answer. And then de novo, well, no, you need to have a primer-dependent synthesis, right? So VPG-dependent priming of synthesis, that's for sure. Circularization, yes. The RNA here is circularized by the poly A PABB interaction. And as I told you, the other class of RNA, plus-strand RNA viruses have a subgenomic mRNA that distinguishes them, but we're not going to go into them in any detail. I want to tell you about the coronaviruses because their replication strategy is pretty crazy. So the coronavirus RNA is 30 kilobases in length. It's present as a nucleocapsid in the virus particle. When the virus fuses, say with the plasma membrane, the RNA ends up in the cytosol. And it can directly engage ribosomes, but those ribosomes will only translate about half of the RNA. And they make these two proteins, 1A and 1AB. Those are basically components of the RNA polymerase. So those proteins end up inducing membranes, and they cause genome replication, so they take the plus and they make it into full-length minus and more plus strands that will eventually be incorporated into virus particles. But you also need to make mRNAs to access all this coding region downstream of 1A and 1AB. And the viruses, all the coronaviruses, make what are called subgenomic mRNAs. They're overlapping mRNAs, each of which encodes one protein, like spike and E and M and nucleocapsid. And these, of course, are structural proteins. They go towards building the virus particle. On the right here is a diagram of these subgenomic mRNAs. So here's the whole genome in B. And when this comes into the cell, only 1A and 1B are translated. And these end up forming the RNA polymerase. So how do you get these proteins made, including spike, right, and nucleocapsid? They're translated from a series of subgenomic RNA. Subgenomic simply means shorter than the full-length genome, right? These are decidedly shorter than the full-length RNA. And each of them can encode multiple proteins, but only one is translated, the very first one. So for example, this mRNA, which encodes 1, 2, 3, 4, 5, 6, 7, 8 proteins, only the 2A protein is made from it. And here, same HE, spike, all the way down here. The only one that encodes one protein is the last one, N, and that's it. So that's how you access all of these proteins that are downstream of the polymerase coding region. And all the coronaviruses do this. It's a universal strategy for accessing this coding region. How it happens is very interesting. Let's see if we can explain it to you. So the RNA polymerase initiates at the 3' end of the genome. Remember, the plus-stranded RNA has come in the cell, it's been partially translated, and you made RNA polymerase. That RNA polymerase now goes to the 3' end of the genome and starts to copy it. And what it does, it will copy to the first, there are a series of intervening sequences called TRSs, they're shown in blue here. The polymerase will go to the first one and then stop, and then the very 5' end of the template folds around, and the polymerase then copies the very end of that, giving it what's called a 5' leader. And this happens for every mRNA. Sometimes the polymerase goes a little further, and that's how you get these nested RNAs. So you're making a series of nested negative-strand RNAs, all of which have a 5' leader sequence derived from the 5' end of the genome from this funny snapback during RNA synthesis. And so then these negative-strand RNAs are copied into plus-strands, which are the messenger RNAs. Now, this is weird, it's Baroque, but the important part here is that this mechanism allows for high recombination rates because the polymerase is moving from one part of the template to another. So for example, when it reaches an intergenic sequence, it jumps all the way to the 5' end. It skips all of this sequence here and adds a 5' leader. So in this case, the polymerase has just synthesized a short mRNA, jumps to the 5' end and picks up the leader. So that movement of the polymerase is what causes recombination. And coronaviruses have high recombination rates. And one of the reasons why they are such a worry, because there's so many of them in nature, and they can recombine to make different viruses. Okay, now moving on to the minus-strand RNA viruses. We're going to look at viruses with segmented genomes, segmented genomes on the right, influenza viruses, and genomes in one piece called unimolecular. So in all cases, the genome is here in the middle, this olive-colored sequence. So for VSV, it's a single negative strand. From that, you make messenger RNA, and you also make plus RNA full length to make more genomes. For influenza virus, same idea. You have a negative strand genome, you make an mRNA, and that you also make a plus strand full length to make negative strands. All right, so let's see how these processes occur. For VSV, of course, the genome is not mRNA, so once the genome comes into cells, it has to be copied to form mRNAs, and that's done by the viral polymerase that it comes in with the virus particle. And each mRNA, which are shown here on the ribosomes, encode different proteins that go towards the synthesis of new virus particles, including the polymerase, which is this protein here called L. Now, when the viral genome is not messenger RNA, there has to be a switch from mRNA synthesis to genome RNA synthesis at some point, when the genome is not mRNA. What do I mean by that? So here again is the genome of VSV. It's a long minus strand RNA. When it gets in the cell, the polymerase initially makes a bunch of small mRNAs. You can't use those to make new virus particles, right? They're too short. They're mRNAs. You need to make a long plus strand and then a long minus strand. So that's what I mean by a switch. When the negative strand first comes in to the cell, the polymerase is making lots of mRNAs. But at some point, it's got to start making genomes to make new virus particles. So that's the switch. What controls it? I'm going to tell you. So here's the genome of VSV here in olive colored. When it gets in the cell, the polymerase, which is attached to it, makes a bunch of mRNAs, N, P, M, G, and L. At some point, this has to be copied to full-length plus strands. The switch is the production of the N protein, the nucleocapsid protein. When the levels of nucleocapsid get high enough, they begin to coat the plus strands as they are made and cause the polymerase to make a full-length plus strand instead of subgenomic plus strands. It undergoes what we call anti-termination, and I believe I have a diagram of that. So when the N proteins reach a certain level, the mRNA synthesis stops and we switch to full-length plus strands, and those are then copied to make full-length minus strands. So that's what I mean by a switch between mRNA synthesis and full-length plus strands. So here's a little detail of what's going on there. So here is the viral genome on the top. And between each gene, between N and P and M, et cetera, there's what's called an intergenic region. So the polymerase initiates at the 3' N and makes an N mRNA. It then stops at the intergenic region. So now you have a complete messenger RNA. And it reinitiates in the P gene. And it makes a P mRNA. Then it stops at the next intergenic region, makes a P mRNA, and then goes into the M and so forth. Okay, so that's the mechanism of making each of these individual mRNAs. So at some point, the polymerase needs to stop this. It needs to stop making small mRNAs and copy the whole thing. And that's the function of the nucleocapsid protein. So this is a detail of the intergenic region now. There's the minus strand genome. There's the messenger RNA that's just been made. The polymerase gets in here, it stops, it reaches this U stretch and begins to chatter and keeps adding a bunch of A's until it falls off. And then you have your messenger RNA. So this is the mechanism of polyadenylation. It reaches this intergenic region. The polymerase begins to slip and copy this U sequence into a poly A sequence. All right, so we have to antagonize that. How do we do that with the N protein? Well, I don't have the picture here, but the N protein will make mRNA synthesis stop and cause the synthesis of full-length plus strands. What it will do is the N protein will anti-terminate in each intergenic region. And so in other words, instead of the N mRNA stopping, when N protein levels are high, the polymerase will continue through P and through M and make a full-length plus strand. I think I have that illustrated for influenza virus, so we can see it there. So again, for flu, it's a very similar story, except the genome is segmented. It's in 8 pieces, but the genome is not mRNA. And again, when the genome is not mRNA, there has to be a switch from mRNA synthesis to genome RNA synthesis. And this is a very complicated slide, but what happens here is that the viral particle gets into the cell by endocytosis. The ribonucleoproteins get out. They have to go in the nucleus, actually. And each of these serves as a substrate for the production of mRNAs, which go out into the cytoplasm and make their proteins. And eventually, when the levels of nucleoprotein get high enough, you switch from mRNA synthesis to full-length plus strand synthesis, and then you get minus strands, which can come out and be incorporated into new virus particles. So again, the switch from mRNA synthesis to full-length plus strands is mediated by the nucleoprotein. So here's the genome, 8 segments of RNA. Each encodes 1 or 2 mRNAs, which encode proteins. So initially, the polymerase, when it comes in the cell with the genome, makes these mRNAs. But I think you can see that you can't make virus particles from those mRNAs, right? They're too short. So how do you make a full-length negative strand? You make a full-length plus strand. Here's the genome in the middle here in this olive color. And it's one segment for clarity. And the mRNA that's made from each genome segment is shown at the top here. Now, the mRNA synthesis for influenza virus is very interesting. First of all, the primer for mRNA synthesis is a piece of host cell RNA with a cap structure. So these are derived from capped cellular mRNAs. And so they end up on the 5' end of the flu virus messenger RNAs. At the other end, the mRNA terminates about 20 nucleotides short of the end of the minus strand. So the mRNAs could never be used to make virus particles. Even if you copied them to a negative strand, they're wrong. The 5' end has got some cellular stuff. The 3' end is missing. So at some point, you have to switch to making full-length plus strands from each segment, and that's shown here. And again, it's regulated by the nucleoprotein. So when you get enough nucleoprotein made, in other words, you're pretty far into the reproduction cycle, you got enough proteins made, now it's time to make virus particles. The nucleoprotein begins to coat the plus strand as the polymerase makes it, and it makes a full-length plus strand. It no longer uses a host cell primer. It doesn't fall short of the 3' end. This is now a full-length plus copy of the RNA, the minus strand genome. And that can then be copied in turn to make minus strands, which will go into virus particles. So these minus strands are full-length copies of the original minus strand. That's why I made the point early on at the beginning that one of the rules is that you have to copy the RNA from beginning to end. You can't leave bits out. Same thing for VSV. I didn't have the slide that showed it, but the VSV terminates at the intergenic region. The nucleoprotein causes anti-termination, so you don't get any small RNAs made. This is the primer for mRNA synthesis. So here's the minus strand RNA. That's the 3' end. And what the polymerase does, it goes to the cell, and the cell is full of mRNAs, and their 5' ends look like this. They have a cap structure, which is basically a G residue flipped around, and then the rest of the mRNA. And so the viral polymerase cuts about 13 bases in from the 5' end and uses that fragment as a primer to make mRNA. So the last couple of bases are complementary to the minus strand, and the polymerase will use this as a primer to make mRNA. So this bottom here with the green is mRNA. So every influenza virus mRNA has a cellular sequence at the 5' end as a consequence. Now there's a new flu antiviral called Xofluza or baloxavir, and it inhibits the endonuclease that cuts the host cell RNAs. That's a viral endonuclease, and someone decided to find an inhibitor of it, and it works very well, actually. If you get flu, ask for Xofluza. It's better than Tamiflu. You only need one dose. You can take it many days after symptom onset, and it seems to work better than Tamiflu. And remember, with VSV, I told you that there's this funny mechanism of polyadenylation where the polymerase gets into the intergenic region and it hits a stretch of U and it freezes and begins to just churn out A's until it puts about 200 A's on, and then the mRNA falls off. Kind of a similar thing with influenza virus. So here's a diagram of the polymerase here. And here in the olive color is the viral RNA, the negative strand RNA. Its 5' end is attached to one of the polymerase subunits shown there. And this other red area is the catalytic site. So what we think happens is that this minus strand genome is pulled through the catalytic site. So the enzyme is not moving down the RNA, but the RNA is being pulled through the catalytic site. So you can imagine the 3' end was originally there, and it's being pulled through, and now you're making a messenger RNA as a consequence. But eventually this loop is going to get smaller and smaller until you can't pull anymore. And guess what? There's a stretch of U's there, and so the polymerase begins to add A's until you get to about 200 A's, and that's how you polyadenylate influenza virus mRNAs. So it's because the template is fixed onto the enzyme, and you get to the end of that loop where you can't go any further. And this also explains why the mRNAs are about 20 bases short of full length, because the polymerase can't copy that last stretch. Just physically cannot. So it adds polyase, and that's why you have shorter mRNAs, and you have to figure out a way to get longer mRNAs. Okay. I know it's a little complicated, but it's not so bad if you think about it. Okay, how are influenza virus and VSV RNA synthesis similar? The switch from mRNA to genome RNA is controlled by an RNA binding protein. Polyadenylation occurs at a short stretch of U. Viral mRNAs are shorter than minus genome RNA. All of the above. How are they similar? All right, let's see how we did. So the answer is all the above, which the majority of you got. So the switch is controlled by an RNA binding protein, totally the same for both. But polyadenylation also occurs at a stretch of U, and the mRNAs are shorter than minus genome RNAs. Yeah, we can look at that briefly here. Let's go back to VSV. So there we have VSV. The genome, the mRNAs are shorter. And polyadenylation occurs as a short stretch of U's in the intergenic region. The polymerase gets stuck and makes a long poly A. Same thing for influenza virus, right? The mRNAs are shorter than the genome. I showed you on this slide that they have a cap at the 5' end, which is wrong, and they're about 20 bases shorter. So they have to be fixed in that way. And plus, the polyadenylation occurs at a stretch of U during mRNA synthesis. So same, all the above. Double-stranded RNA viruses. Can double-stranded RNA be translated? No, good. It will serve you well. Cannot be translated. Just think, it's so twisted up. The ribosome cannot get to it. I mean, there's a plus strand there, but the ribosome cannot access it. All right, so the ribosomes can't access double-stranded RNA. So what has to happen? These viruses have to have an RNA polymerase in the particle, which will make mRNA from the double-stranded RNA. Basically, what they will do is copy the minus strand and make an mRNA. And that can be used to make proteins. And eventually, it can be used to make more virus particles. For these viruses, the mRNA and the plus strand that's incorporated into this double strand is exactly the same. So there's nothing different there. So let's see how this happens. This is a very cool process. Here's the virus particle. Remember, it has a double icosahedral shell. It has an outer shell, and then it has an inner shell. And in the process of this virus entering the cell, the outer shell is stripped off. These plugs are removed from the turrets. And this particle, the core, is very hydrophobic, so it punches through the endosome and ends up in the cytosol. All right? And it contains, multiple double-stranded RNA segments, as you can see here. So that's shown here. The particle is taken up by endocytosis. The outer shell is stripped off, and you end up with a core in the cytosol that has the outer shell completely stripped off, but of course is full of double-stranded RNAs. Those double-stranded RNAs stay in the particle. They never leave. So this is one example of a virus where the genome never leaves the particle. They stay in there, and the polymerase makes mRNAs from each of the segments, 10 or 12 or whatever you may have. And that's shown here, these little squigglies coming out of the turrets. Those are mRNAs being made by the polymerase that is inside the core particle. In fact, we think there's a polymerase at every turret. Each turret is at the 5-fold axis. We think there's a polymerase at each turret so that it can copy the RNA that's below it and make mRNA. So those mRNAs come out into the cytosol. They make proteins. You can make new virus particles. You can assemble, you can put the mRNAs into the virus particles. You can make them double-stranded. You can add the outer shell, and you have new virus particles. So the viral genome is not mRNA. So where is the switch to genome synthesis? Well, the switch to genome synthesis is right here. So initially, we're making mRNAs, and they're coming into the cytosol. They're being translated. They're making new proteins. So here, the first step here is 8. We have a core with single-stranded RNAs. When that gets double-stranded, that's the switch to genome synthesis, because that's what double-stranded RNA is, a genome synthesis. whereas before that, we're just making mRNA. So this is a very cool entry scheme. First, that the particle can be digested so it penetrates into the cytosol, but also the RNA stays inside and never leaves. I think that's very cool. Now, here's a neat experiment that was done years ago where they took the core particle of, in this case, rotavirus, but same double-stranded RNA virus, and they incubated them in vitro with triphosphates, and they made electron micrographs of the particles. And you can see the mRNA is coming out of each turret. So each of these is an mRNA. It's got a cap at the 5' end, that's the red structure. And so each turret, which is a 5-fold axis, is extruding an mRNA. So there's an RNA polymerase right below the turret, and that's copying the double-stranded RNA and making mRNA. Is that cool or what? It really is, isn't it? I think it's great that it's just sitting below there. And guess what? How many 5-fold axes are there in an icosahedral particle? Do you remember? 12. Do you know the maximum number of double-stranded segments there are in these viruses? 12. You can't have more than 12 because you have one segment per 5-fold axis, because that's where the polymerase sits. You can have less, you have 10 or 8 or 6 or whatever, but you can't have more than 12. Okay, last thing I'm going to talk about is making mistakes, origins of diversity. All viruses misincorporate nucleotides. Remember, I told you that the 4 triphosphates just go in and out of that nucleotide entry channel until the right one is picked. And sometimes the wrong one is incorporated. So both DNA and RNA polymerases do that, but RNA polymerases don't have error correction mechanisms. So they make way more mutations than DNA viruses. We'll actually compare them later on. But RNA polymerases don't have proofreading activity, and so they make a mistake every 1,000 or 10,000 bases polymerized. One mistake. So you have a genome of 10,000 bases. Every time it's copied, you get one mistake. If you make 10,000 genomes, all the mistakes are there. Every base has been theoretically changed. So there it is. In a 10 kb genome, a mutation frequency of 1 in 10 to the 4 results in 1 mutation per genome. So this is why RNA viruses make a lot of mistakes and why they vary so much. Now, the exception of the Nidovirales, the order which includes the coronaviruses. They have an error correction protein called XON, which corrects RNA polymerase errors. If you take out an XON gene from the virus, you make a virus that makes more mistakes, as you might guess. And why do the coronas have XON and not other RNA viruses? Well, they're the biggest RNA genome. So except for outside of the coronas, the biggest are 10 to 12 kb. Coronas are 30, 40 kb and higher. They're really substantially higher. And we think without an error correction enzyme, you cannot sustain such a big genome. You would have too many errors and it would make mutations that would be non-infectious. So we think this is there because of that, to allow replication of large genomes. But they're still small compared to DNA genomes, and that's probably because RNA is also fragile on top of having a high mutation frequency. Okay. Now for poliovirus, people have studied what controls the fidelity of its synthesis. And this is, so here we have a structure of the active site. There are your yellow residues, the 2 asps. And here is a uridine, I believe, uridine triphosphate that's in the active site. So it's about to be added to the growing chain. And how this interacts with the enzyme determines the error rate, basically. So the NTP first binds in a way that doesn't allow the ribose to interact with the acid amino acids. But if it's correctly base paired, we'll have a conformational change in the enzyme, reorienting the triphosphate, and that allows phosphoryl transfer to occur. So in other words, if the right triphosphate is in, you get a conformational change in the enzyme, which now allows it to be incorporated, which wouldn't happen before. You can make changes, single amino acid changes in the polio polymerase that affect the error rate. So here's the polio polymerase on the right. There's the active site in yellow. And on the back, this single amino acid 64, if you make a change from G to S, the enzyme makes fewer errors. What it does is slows the conformational change that happens on base pairing. It reduces the elongation rate. And this is interesting because it's in the fingers domain. It's a remote from the active site, but it makes the enzyme more dependent on correct base pairing of the NTP in the active site, and that's why it increases the fidelity. And this seems to be conserved among other RNA polymerases as well. However, as we'll see when we talk about evolution, these enzymes suck in nature. They cannot survive. If you put them in a competition assay, they lose. Mutation is selected for in nature. That's how you get variation. So that's fidelity control. There's another aspect of mutation in plus-strand viruses, which is RNA recombination. And that's exchange of nucleotide sequences among different molecules. This is totally distinct from reassortment. So here we have a cell infected with two viruses, two different plus-strand viruses, one green, one hatched green. And so the RNAs are both being copied in the same cell. And from time to time, the polymerase will start copying one RNA and then switch to another, with the result that you have a recombination event occurring. This is very, very high frequency. For poliovirus, up to 20% of RNA molecules will recombine in a single replication cycle. And this has big implications for the vaccine strains, which we will talk about later. And so RNA recombination is another important mechanism of genome plasticity, not just mutation rate, but the ability to exchange fragments. Interestingly, negative-strand viruses don't do this. There's no evidence for recombination among them. I'm not sure why. And for poliovirus, we've identified, not we, but the field has identified 4 amino acids in the polymerase that control recombination frequency. And these amino acids are shown here. They are in the thumb domain in the RNA exit channel, actually. So here's the active site again. They would be in the RNA exit channel. And they contribute to the primer grip, the ability of the enzyme to grip the primer and distinguish between homologous and non-homologous template. If you make changes at these amino acids, you reduce the recombination rate. And I will tell you a really interesting story later when we talk about vaccines, about how individuals tried to combine fidelity control with recombination control to reduce reversion of a vaccine strain. And the moral of the story is don't try and fight nature. You're always going to lose. Next time, we're going to talk about making mRNA and processing it from viruses with DNA templates.